Hi, my name is Ryan Powell, and I'm here to talk to you about simple and scalable cell therapy. Now, every human starts as a single cell, which then grows and divides into more than 37 trillion cells. If something goes wrong during that process, and a cell begins to grow and divide uncontrollably, then you get cancer. Layla here has an amazing story. She was born with an untreatable blood cancer known as acute lymphoblastic leukemia. After the doctors tried everything, they infused her with genome-edited cells from another patient, and those cells reversed her cancer. Now, this is great for Layla, but there are literally millions of people dying every year who could use those genome-edited cells. So let's take a step back. In order to engineer one of these cells, you need a functional molecule, like CRISPR-Cas9, and you need to deliver that molecule across the cell membrane and into the cytosol. Right now, there are two big ways to do this. One, by inducing a pore using a technology called electroporation, or two, with a vector such as a virus. Now, dying patients are getting turned down by hospitals because the manufacturing scale of these therapeutics is limited to thousands of patients per year. So manufacturing these therapeutics at scale is the problem. If you look at cancer alone, 8.2 million people die every year. So here's what key opinion leaders have to say about viruses and electroporation. Calimmune is developing an anti-HIV cell therapy using viruses, and Westmead Hospital is the largest bone marrow transplant in the Asia-Pacific region. Calimmune needs a user-friendly way to do this, and the director of that hospital uses electroporation, but thinks there are no good options here at present. So our microfluidics represent the ideal solution. These devices can be manufactured at scales of 10 million per year, which is more than enough for the 8.2 million people who die every year of cancer, and orders of magnitude greater than the current manufacturing scale with viruses or electroporation. So here's how it works. You mix your molecules and your cells in suspension, you flow them through our device, and on the outlet, you get your molecules inside of your cells. So we use features that are 1/10 to 1/100th the size of a human hair, and we flow our cells and molecules past them at more than 100 miles an hour. Our patent-pending fluid dynamics then disrupt the cell membrane, allowing for molecule delivery. We started Indee Bio with an idea and some basic proof of concept data. Since then, we've gone on to demonstrate delivery with long-term immune model survival, and are even generating the Cas9 protein inside these cells, which is a proof of concept for the therapeutics that were used to reverse Layla's cancer. So we're not the only ones working on this problem, and here's how we stack up against our competitors. Squeeze Biotech also uses microfluidics, but our yield is 5-fold greater. Our cell viability is nearly double. Now that's important. You don't need a lot of cells, according to Professor Carl June, or the godfather of adoptive immunotherapy, but you do need enough to induce a therapeutic effect. And if you infuse cells with less than 80% viability, regulators won't like that. Why? Because you can cause an adverse immune response. We've also found that the market likes Lonza's electroporation system, but we aren't worried about that because our numbers are better. So here's our plan. Right now we're optimizing our delivery technology, and then we're moving towards gene editing. At the same time, we're also focused on a cloud lab or lab automation, as that will allow us to turn out high volumes of data. After that, we're focused on partner deals, and then we'll move towards an FDA device master file, which means we'll be able to manufacture these therapeutics for the clinic. We have a substantial competitive advantage. We have a PCT filing. Our technology is scalable. We can make more than 10 million devices a year with existing manufacturing capabilities. We have high margins. Our chips cost less than $20 to make at an R&D scale, and it costs more than $6,000 for enough viruses just to treat one patient. We've also minimized risk. I use medical materials and approved suppliers, and we also have proprietary quality control software. So here's our business model. Our expertise and the highest margins lie in therapeutic development. We anticipate about $20 million in development milestone payments for each target. On the back of that, we hope to get about at least $30 million per rare disease in device sales alone. Now, every cell consists of an intracellular environment that's surrounded by a phospholipid bilayer or cell membrane. That means we can access any market that utilizes cells, including industrial and agricultural biotech. So here's our traction. We're talking to multiple big pharma companies with market capitalizations up to $200 billion. We're talking to 2 large gene editing companies, and we're also talking to a profitable cell therapies company, plus a big ag biotech company. Now, we can't put their names on there because we respect our confidentiality agreements. However, we're at the evaluation stage with a few of them. So here's our team. Matt is a weapon. He's got more than 10 years of experience in cell biology and disease modeling, and I specialize in microfluidics and medical devices. We're backed by a series of advisors, much who've been providing advice and mentorship for over half a decade. Also, some of them specialize in billion-dollar outcomes. Todd Martin, for example, helped draft and prosecute the $1.35 billion Mickelson patent portfolio, and Mike Bowles led a startup to a $3 billion IPO after just 2.5 years. So thank you very much for your time. We've already raised more than $500,000, but we look forward to talking to anybody who is willing to support our efforts.